Researchers at the Tafaris Lab, University of Oxford, have devised a blood-based test to identify individuals at high risk of developing Parkinson's disease. Parkinson's disease begins decades before symptoms appear, when the processing of the protein alpha-synuclein inside nerve cells fails, forming clumps that cause cell damage. In people with Parkinson's disease, small amounts of alpha-synuclein are packaged into tiny particles called extracellular vesicles, which are then released into the blood. Extracellular vesicles are naturally released into the blood by all kinds of cells from different organs in the body. The researchers have developed an antibody-based test designed specifically to capture those extracellular vesicles that originate from nerve cells. The test uses composites of tiny magnetic particles called nanobeads, which are coated with antibodies that specifically bind to the vesicles that originate from nerve cells. In this way, these vesicles can be extracted by magnet and tested for alpha-synuclein. This overcomes the previous limitation that more than 99% of alpha-synuclein in blood comes from peripheral sources that are not relevant to the disease process. Increased alpha-synuclein levels in these vesicles indicate failure to process the protein inside nerve cells and higher risk of Parkinson's. Early identification of this risk could improve diagnosis, clinical trial design, and the timely initiation of targeted therapies when they become available. This laboratory test could eventually be developed into a clinical screening test for Parkinson's disease, such as we currently have for some common cancers.